Welcome to the deep dive. Today we're taking a really exhaustive look at one of the most stunningly complex feats of human embryology, the development of the respiratory system.
It's a fantastic topic.
We're going deep into the anatomical source material to understand how the lungs, the thorax, and the diaphragm all get built.
And when you study this, you realize just how delicate it all is. I mean, it's not the growth of a single organ. It's more like a massive, synchronized construction project.
The sources we're looking at have a great way of putting it. They call it the growth of 6 intertwined trees.
That's right, 6 trees.
6 trees. That sounds like an entire forest being built inside a fetus.
Mm-hmm.
So, what are we tracking here?
Well, you have the 2 core functional trees.
Right.
First, the conducting bronchial tree. That's basically the pipes that move the air. And then you have the alveolar sacs, where the gas exchange will eventually happen.
Okay, the airways and the air sacs.
But those need plumbing. So you also have 4 vascular trees, the systemic, arterial, and venous trees, which are, the nutrient supply for the lung tissue itself.
Right.
And then the absolutely critical pulmonary, arterial, and venous trees, which are for gas exchange after birth. And finally, the lymphatic tree for drainage.
That is just immense coordination. And I think this brings us to the core paradox, the thing that makes this deep dive so compelling. The lungs spend 9 months developing without ever doing their job, oxygenating blood. Yet they have to work perfectly instantly the second the umbilical cord is cut.
Precisamente. If any one of those 6 trees, or the chest wall, or the diaphragm is misaligned, the consequences are immediate and severe.
So our mission today is to trace this whole process chronologically. We're going from the first buds to the adult architecture. And we'll highlight the clinical weak points along the way to give you a complete shortcut to understanding this masterpiece.
Let's do it.
Okay, let's unpack the embryonic origin, the absolute starting point. Where does this all begin?
It starts remarkably early. We're talking stage 12, so only about 30 or 31 days post-fertilization.
Wow.
The lungs originate as what's called the respiratory diverticulum. It's essentially a ventral bulge, little pouch, that sprouts from the endodermal foregut.
And the foregut is that primitive tube that will become the esophagus and stomach.
Exactly. And this little bud pushes out into the surrounding tissue, which is called the splanchnopleuric mesenchyme.
That is a mouthful. Splanchnopleuric mesenchyme. So what's its job, simply put?
It's the command center, the actual lining of the airways, the epithelium that comes from the endodermal bud. But that mesenchyme around it, it provides all the connective tissue, the cartilage, the smooth muscle, and crucially, all the instructional signals that guide the growth.
So the very first challenge is separating the breathing tube from the eating tube.
Absolutely essential. The trachea and esophagus start to separate from that common origin. We tend to think of it as, zipping up from the bottom.
Yeah.
But the process is actually defined by the distal trachea lengthening. The original opening stays put, but the whole lung system stretches down, pulling the future branching point, the carina, deep into the thorax.
And if that separation fails to zip up completely, that's where we get into trouble.
That's when we see a tracheo-oesophageal fistula.
Correct.
And that's pretty common, right?
It's the most common major abnormality of the lower respiratory tract. You see it in about 1 in 3,000 births. And these fistulae, these abnormal connections, they're often not isolated.
Okay.
They're frequently associated with the VAC-trial complex. That's a whole group of co-occurring anomalies involving the vertebrae, anus, cardiac structures, trachea, renal system, and limbs. It just underscores how linked everything is at this early stage.
Okay, now let's move to the branching of the airways, this dichotomous branching. What's so surprising here is the total dominance of that mesenchyme you mentioned.
It's the director of the entire symphony. It dictates the pattern. And we know this from some pretty dramatic experimental evidence. If you take bronchial endoderm, which naturally wants to branch, and you surround it with mesenchyme taken from the trachea, the branching is completely inhibited.
So the tracheomesenchyme has some kind of anti-branching signal? It does. And even more amazing, if you take rat fetal airways and expose them to chick mesenchyme, the rat airway starts developing a chick-like branching pattern.
That's incredible. So the blueprint is entirely external. It's in the surrounding tissue.
Exactly. The structure isn't determined by the lining, but by the physical instructions provided by the connective tissue matrix around it.
And there are even molecular signposts, little chemical flags telling it where to go.
Yes, the extracellular matrix. You have tensin, for instance, which is always found right at the budding tips, sort of inviting forward growth. And then you have fibronectin in the clefts and along the sides, which establishes the boundaries and keeps the tubes separate.
Okay, so let's shift from that initial structure to the chronological maturation. Anatomists break this down into a few distinct phases based on what the tissue looks like.
Right. This helps us map the timeline of readiness for birth. We start with the embryonic phase, from weeks 0 to 7. This is the period of the initial budding and growth. The lung buds push out into these spaces called the pericardio-peritoneal canals, just lateral to the esophagus.
And this whole phase is governed by some pretty powerful genetic switches.
It is. A key regulator here is a transcription factor called NKX2.1. Clinically, if there are mutations in NKX2.1, children can present with a combination of lung, brain, and thyroid diseases.
Because that one factor is critical for all three.
It governs early development in all three of those seemingly unrelated organ systems.
I see. Next up is the pseudoglandular phase, from week 5 to 17. Why pseudoglandular?
Because under a microscope, the dense packed tubes look just like an exocrine gland. This phase is all about laying down the conducting network. By week 16, pretty much all the conducting airways are formed. That's about 20 generations of branching. Done.
So the pipes are all there.
The pipes are there. You start to see differentiation, 2 ciliated cells around week 7, mucus glands by week 12. But, and this is critical, these airways end in thick, blind-ended sacs. No gas exchange is possible yet.
Which changes dramatically in the next stage, the canalicular phase, weeks 17 to 27. And this seems to be the true watershed moment for survival outside the womb.
It's the point where the blood-gas barrier actually forms. The term canalicular refers to the widening air spaces, or canals. The central event here is a massive decrease in the mesenchyme between the airways and the blood vessels.
The packing material goes away.
It shrinks away, and the distance between the airway lining and the capillaries thins out dramatically.
And this shrinking tissue triggers cell differentiation.
Exactly. The primitive cuboidal cells change into 2 new types. You get the type I pneumocytes. These are super thin, flattened, and cover about 90% of the surface. They're the gas exchange surface.
And the other type?
The type 2 pneumocytes, they're still cuboidal. They store and secrete surfactant. That's the detergent that keeps the air sacs from collapsing. And they also act as stem cells. They can regenerate the lining.
So the ultimate goal of this phase is getting the capillaries right up against those thin type I cells.
Intimate apposition. That's the key. Before that happens, there's no hope of gas exchange. If a baby is born before this phase is well underway, they just don't have the structure to breathe air.
If they get past that stage, we move into the saccular and alveolar phases. This starts around 28 weeks and, well, it continues long after birth.
That's right. The saccular phase is defined by these thin-walled terminal saccules, but the key event is the formation of secondary crests.
Secondary crests.
Imagine the wall of a saccule starting to have these small folds or invaginations pull inward. This is driven partly by elastin fibers being laid down, and these crests start dividing the big saccules into smaller, primitive alveoli.
And the massive expansion in the number of these alveoli, that's mostly a postnatal event.
Vastly postnatal. The number at birth is debated, but alveolar formation just explodes, especially in the first 6 months of life. And get this, the final stage, the fusion of the double capillary network between adjacent alveoli, that's often not even complete until a child is about 5 years old.
So development is a very long-term project.
It really is.
Let's pivot now from the air passages to the plumbing,
Yeah.
the vasculature. This is where fetal and postnatal life just diverge completely.
It's a critical distinction to make. You have 2 separate circulatory systems.
Okay.
The bronchial circulation supplies the lungs' structural tissues, the cartilage, smooth muscle. It's a systemic circulation coming off the aorta. It carries nutrients.
And the other one.
Then there's the massive pulmonary circulation. This one is all about gas exchange. It originates from the 6th aortic artery.
In utero, that pulmonary circulation is under incredibly high pressure. High pulmonary vascular resistance, or PVR. Why so high?
It's a structural thing. Early on, the pulmonary arteries get a really thick muscular wall. This thick constrictive layer means only about 10% of the cardiac output actually goes through the fetal lungs.
The rest is shunted away.
Exactly, through fetal bypasses. The pulmonary veins, on the other hand, develop just a single thin layer of smooth muscle. They're built for low pressure.
So this high resistance is built right into the fetal structure. But the lung isn't just passive, is it? It's actively making fluid and exercising.
That's right. The fetal lung is a net fluid secretor. It's constantly pushing out fluid, which keeps the airways distended. And you have fetal breathing movements, rhythmic diaphragm activation.
Even though it's filled with fluid.
Yep. These movements are essential. They mechanically stretch the lung, which stimulates the release of growth factors vital for development.
Which brings us to a really important clinical point. Oligohydramnios, insufficient amniotic fluid.
Yes, you often see this in conditions like renal agenesis as part of Potter's sequence. If the amniotic fluid volume is too low, there's not enough pressure around the fetus.
And that pressure is needed for the lung to expand.
It's vital. Without that necessary distension stimulus from both the outside pressure and the internal fluid secretion, the result is pulmonary hypoplasia. The lungs are just too small and underdeveloped. It's usually fatal.
It really shows how dependent lung growth is on these external physical forces. Okay, let's look at the container for the lungs, the thorax and the diaphragm.
Right. The chest cavity starts as part of one large space. The lungs bud into what are called the pericardioperitoneal canals, so we need to wall those off from the heart and the abdomen.
Let's start with the top, separating the lungs from the heart.
That's done by the pleuropericardial membranes. They grow in from the sides, and they carry the common cardinal vein and the phrenic nerve with them. They fuse, and you get your separate pleural and pericardial cavities.
And then closing it off from the bottom, from the abdomen.
That's the job of the pleuroperitoneal membranes. What's important here is the asymmetry. The right side closes earlier than the left.
And that's why...
That's why when you see a diaphragmatic hernia, a persistent communication, it's far more frequently on the left side.
Now, the diaphragm itself, you mentioned it's a mosaic. It's built from multiple parts.
4 key mesenchymal contributors. The main central part comes from the septum transversum. Then the pleuroperitoneal membranes close off the sides. And the very back parts, the deep recesses, they come from the somatopleuric mesenchyme.
It's like 4 different contractors building one floor. But its descent is maybe the most incredible part.
It's dramatic. The muscle cells that form the diaphragm actually originate from cervical somites C3 to C5, up in the neck.
In the neck.
As the embryo lengthens, the diaphragm gets dragged all the way down to the thoracic lumbar junction, pulling its nerve supply, the phrenic nerve, along for the whole ride.
Which explains why a nerve from the neck controls a muscle deep in the chest. And if those posterior parts fail to fuse, we get the most common defect.
The posterolateral Bach-Delitzsch hernia, correct, accounts for 85 to 90% of cases, almost always on the left. And this is where our clinical understanding has really shifted.
How so?
Historically, we blamed the hole, the failure of fusion. But there's more and more evidence that the primary problem is actually lung hyperplasia, abnormal growth. The herniation of the gut into the thorax seems to be a secondary event that just makes a bad situation worse.
A chicken and egg problem where the underdeveloped lung comes first. Okay, let's get to the final life-altering moment.
Yeah.
The first breath. That PVR has to drop instantly.
It's a massive shift from high to low PVR, and it uses 2 mechanisms at once. First, mechanical. The chest wall expands and literally pulls the pulmonary vessels open.
And second,
chemical. Oxygen floods the alveoli for the first time, and that induces a massive pulmonary vasodilatation. The combination of those two is what switches the circulation over.
But that newborn system is incredibly volatile.
Highly vulnerable. If the neonate becomes even slightly hypoxic or acidemic, which is very easy right after birth, that high PVR can spike right back up. The fetal shunts can reopen, and you get what's called persistent fetal circulation. It's very dangerous.
And the physical structure of the chest itself is still immature.
Yes. The neonatal chest is rounded, not flattened like an adult's. And critically, the chest wall is up to 5 times more compliant than the lungs. The lungs are stiff, the cage is soft.
And that's why you see that classic sign of distress.
Exactly. If the baby struggles to breathe, the chest wall indraws. It collapses inward with every breath because the cage is just so deformable.
And finally, that arterial remodeling, it continues for years.
It's a long 3-stage process. Stage 1, the first 4 days, you get a dramatic reduction in that thick, smooth muscle. Stage 2, up to 4 weeks, they lay down the elastic lining. But stage 3, the full maturation of that muscular layer can take up to 2 years.
And interrupting that sensitive window can cause permanent damage.
Absolutely. If alveolar development is arrested like in very preterm infants on ventilators, it's a major factor in bronchopulmonary dysplasia. The harm can be irreversible.
It is truly staggering to realize that every single component, the air sacs, the high-pressure arteries, the thin veins, the diaphragm, the chest cage, all have to perfectly coordinate their maturation just to enable life outside the uterus.
It really is the ultimate testament to anatomical coordination. The switch from being a fluid secretor to an air absorber, alongside the remodeling of the circulation and the sealing of the diaphragm, that's the foundation for understanding almost all of pediatric respiratory pathology.
And as we close, Here is a final provocative thought from the source material. We know that the first 4 to 6 years of life are a key window. After that, there's effectively no catch-up growth in lung function. Knowing how vulnerable this developing architecture is, what does this mean for children exposed to chronic insults like air pollution or passive smoke? And how does that permanently set their respiratory capacity for the rest of their adult lives?
That's a critical question. It reminds us that the journey to a mature, resilient lung lasts far, far longer than that first complicated, life-saving breath.
Thank you for joining us for the deep dive.